Good morning. I am Dr. Egidius James Rwabuga from the School for Field Studies, SFS, Tanzania Center. The School for Field Studies, or SFS, is the United States' largest environmental study abroad program provider for undergraduate college students, offering fully accredited semester and summer-long academic programs in 12 countries around the world. SFS students and staff conduct field research driven by strategic research plans to address environmental issues which affect both the communities in which the organization operates and the world in general. More than 18,000 students have studied abroad with SFS since it was founded in 1980. SFS is a non-profit organization currently based out of Beverly, Massachusetts. SFS offers more than 20 semester and summer programs around the world. Programs are based out of SFS operated permanent field stations, which are located in areas of opportunity for academic study and community engagement. The field stations, known as centers, are home to faculty, staff, and students, and anchor the strategic long-term research upon which the SFS mission is based. Each SFS center has a particular academic focus and set of research themes to reflect the surrounding ecosystems and environmental issues. Semester program coursework generally covers ecology, environmental policy, natural resource management, and socioeconomics, while summer programs focus more on specific topics. In Tanzania, the SFS Center for Wildlife Management Study is located at Rorya in Karatu District, Tanzania, near Lake Manyara National Park and the Ngorongoro Conservation Area. Programs offered include wildlife management and carnivore of the African plains summer programs. Research and coursework explore themes related to wildlife management and ecology, human-wildlife conflicts, national parks management, and climate change. The School for Field Studies programs Tanzania take place in world-famous national parks, wildlife sanctuaries and protected areas, and indigenous communities and small villages. Courses and field work focus on key environmental issues faced here, such as biodiversity conservation, wildlife management, human-wildlife conflict, tourism impacts, and climate change. Tanzania has made progress in protecting its large populations of charismatic wildlife, but this rich landscape faces many challenges. Our research concentrates on understanding the impacts of climate change, habitat fragmentation, and competition for resources, and promotes successive coexistence between humans and the country's incredible wildlife. Hence, research themes include wildlife conservation, climate change impacts, African large mammal behavior and ecology, feed ornithology, human-wildlife conflict, habitat assessments, community-based conservation, and reptile ecology and conservation. Also, students engage in direct uh, res- uh, research projects. This project prepares students to distinguish hidden assumptions in scientific approaches and separates fact from interpretation, cause from correlation, and advocacy from objectivity. Students learn specific tools including experimental design, fit techniques, basic descriptive statistics, and parametric and non-parametric quantitative analysis. Emphasis is placed on succinct scientific writing, graphic and table representation results, and effective delivery of oral presentations. Furthermore, here at SFS Tanzania Center, we are running a long-term small mammal mountain project. Why small mammals? This is because historically, research on large mammals has been given a lot of attention as compared to small mammals. This has resulted into inadequate information on various ecological aspects of small mammals that are essential for their conservation and management. However, there are, no, there are so many species of small mammals on the continent, on the African continent, and elsewhere, especially categorized into five uh, orders: Rodentia, Insectivora, Scandentia, Lagomorpha, and Chiroptera. Of the five orders, the majority of small mammals are rodents, which are rats, mice, voles, squirrels, etc. With more than 2,000 species recorded globally, but comprising 73% of all mammals in Africa. Out of the 2,000 small mammal species recorded, rodents are about 380 species, whereas the bats are around 200 species. Hence, the sheer number of small mammal species and the large population numbers of some of these species uh, deserve substantial recognition as they are particularly important because of their multiple functional roles in the ecosystem. The animals occur in virtually all habitats. Their feeding habits include grass eaters, seed eaters, carnivores, insectivores, and fruit eaters. Their activities result in cycling of nutrients, and they have many beneficial interactions with plants such as seed dispersal, seed germination, and pollination of flowers. Small mammals modify local environments, either ecological engineering, by their burrowing activities, and they provide food for many predators. Some species are reservoir hosts for microorganisms, which cause diseases in humans and domestic animals, and others are pests in agricultural and stored products. Birds are of value as predators of insects, and some species of rodents are utilized as food by humans. Thus, small mammals have roles in most ecological systems and in all habitats, and many of their activities impinge on other animals, including humans. Let's now talk a little bit more about rodents, which seem to be the most captured in our small mammal mountain project at our center and have a bad history in villages surrounding it. As it has been pointed out earlier, rodents contribute about 42% of the mammal species. They are known to survive in a variety of habitats from swamps tropical forests to deserts. There are various ecological aspects, including population dynamics, reproduction, growth, and survival, are linked to ecological factors such as seasons, climate, competition, habitat, and rainfall patterns. They are ecologically important to ecosystems such as seed predators, ecosystem engineers, nutrient cyclers, and prey species, as well as socioeconomic impact on humans, as some rodent species are a source of protein for humans. Generally, it is well known, however, that natural habitat modification as a demand for agricultural land and settlement affects small mammal populations and distributions. This is happening in many Africa's protected areas in the tropics as they are progressively becoming vulnerable to human encroachment, which is featured by anthropogenic land use and land cover changes, which have altered breeding and other population demographic parameters of small mammals, including rodents. Alterations to natural habitats by land use can modify the constraints imposed by environmental structure and seasonality on the timing, synchrony, and fecundity of breeding by small mammals. The relationship between small mammal species population dynamics and community species diversity, human crop depreciation, human crop predation, and transmission of zoonotic diseases is well documented. For example, birth, death, immigration, and migration are the four primary ecological events known to influence the dynamics of a population. The four parameters can be formulated into a simple equation that expresses the phenomena. Change in population density equals birth plus immigration minus death plus immigration. All other factors, either biotic or abiotic in nature, are considered as secondary and exert their impact on population density by influencing one or more of the variables on the right-hand side of the mentioned equation. Such factors are broadly categorized as density independent or exogenous factors, e.g. volcano eruption, and density dependent or endogenous factors, e.g. competition. Endogenous factors cause changes in a dynamic variable, e.g. the per capita growth rate, which is in turn affected by those changes. In contrast, exogenous factors may influence the per capita growth rate that are not affected in turn by those changes. Further, influences on population fluctuations can occur synchronously over large regions due to a response to some region-wide processes, or of a small scale on small localized areas, suggesting the contribution of local rather than regional processes. Changes in diversity and abundance may lead to negative impacts on conservation and public economy. For example, exclusion of specialist rodent species from communities may give rise to higher population sizes of pest species. For instance, the multi-mammoth mouse, Mastomys natarensis, is an important pest across southern and eastern Africa that causes substantial crop losses. In Tanzania, M. natarensis has been labeled as the most important rodent pest that can damage up to 40-80% of farmers' feed crops in as low as 18 individual animals in hectare in a hectare of crop farm. On the other hand, reduced diversity may increase spread of zoonotic diseases due to compromised dilution effect, as rodents are host of several ectoparasites which act as vectors of pathogenic microorganisms and parasitic of zoonosis such as Yersinia pest bacteria causing plague. Any change in the ecological system that affects rodents hosts, it also affects ectoparasites through a mechanism that is well explained by interactions of host animals and ectoparasites between habitats and climate. For example, land use change and climate affect the risk of plague transmission by influencing Yersinia pest bacteria, free vector, and rodent host development, behavior, and survival in affected areas. Plague is a zoonotic disease causing a major public health globally. It is transmitted by fleas infected with bacteria Yersinia pest after taking blood meals from infected animals. The disease has been reported in many mammalian species, but rodents are the most important host that harbor fleas responsible for maintaining the disease. The disease is still active in African countries including Madagascar, Mozambique, Uganda, Republic of Congo, and Tanzania. Between the year 2014 and 2015, more than 90% of the worldwide cases were reported. Madagascar and Republic of Congo were the highest endemic countries. In the recent plague outbreak in Madagascar, a total of 2,348 cases were confirmed as plague suspects, with 202 human deaths recorded. In Tanzania, plague can be explained as a emerging disease since it has been on and off since pre-colonial times. The largest plague outbreak occurred in Shoto, eastern Tanzania in the year 1980, claiming 640 human lives and persisted for 13 years. This was followed by other outbreaks in Karatu and Bur district in the year 1996. In general, records show that most fossa in Tanzania have experienced several outbreaks of the disease, whose transmission is influenced by various factors such as climatic conditions, temperature, humidity, and rainfall. Multiple vector interaction with the host accelerates disease persistence in the active fossa. Also, social, cultural, human behavior and lifestyle practices in the communities increases disease transmission in the communities. As it has been mentioned earlier, Karatu district, where our center is located, is one of the plague foci found in Tanzania. Hence, understanding the ecology of disease transmitting agents is necessary. That is one of the reasons we have established a long-term small mammal trapping project at Moyo Hill, the SFF's Tanzania Center. The aim of the project is to investigate the ecology of small mammal species and free parasites in Karatu district for a prolonged period of time. Results of this study will provide information that primarily can be used for proper management of zoonotic diseases within the study area and elsewhere. Also, The project is due to the fact that a number of studies have been conducted in the Karatu district to investigate aspects responsible for zoonotic diseases transmission associated with the small mammals. In the year 2022, Sonora and others investigated antibiotic resistance, AMR, and virulence profiles of Staphylococcus aureus and Escherichia coli from rodents, humans, and chicken coexisting in Karatu Desert. Results of this investigation revealed high frequencies of isolation for S. aureus and E. coli in rodents, humans, and chicken and soil samples. It has been reported that interaction of rodents with humans and livestock and households environment is rampant in Karatu districts and is believed to be facilitating widespread of many disease-causing germs, including resistant bacteria infections among different hosts in the community. Worldwide, there is a major concern that antimicrobial resistance is a rapidly growing multifaceted problem which threatens global security, public health, and the economy. Currently, about 700,000 humans worldwide lose lives annually due to AMR infections that are difficult to treat, which are also associated with high health care costs, elongated time spent in the hospitals, and increased animal production costs. Therefore, it is recommended that enforcement of rodent control measures should be effected, including biological control, that is use of cats or cat urine to scare rodents, physical, that is by trapping them, and chemical, that is by using toxins, or a combination of them to ensure that rodents have limited contact with humans. Also, Issa and others in the year 2003 explored pathogenic and zoonotic bacteria from wild rodents, dogs, and humans in Ngorongoro Conservation Area, a few kilometers from our center, whereby several zoonotic or airborne contagious bacteria, including Mycobacterium species, Mycoplasma species, Borrelia species, and Legionella species, were detected in wild rodents, domestic dogs, and humans. Arthropod-borne zoonotic bacteria such as Bartonella species, Borrelia species, and Kestia species were detected in all three hosts, while Orientia species was found in wild rodents and domestic dogs. Yersinia pest, Streptobacillus species, and Anaplasma species were found only in wild rodents. Other zoonotic bacteria found shared among wild rodents, domestic dogs, and humans are Leptospira species, Brucella species, and Salmonella species. Generally, wild rodents have the highest prevalence of zoonotic bacterial species when compared to domestic dogs and humans. Hence, it was concluded that detection of zoonotic bacteria in rodents, dogs, and humans supports the hypothesis that infections can spread between animals and humans sharing the same environment. Therefore, it was recommended that there should be sustained investigations and unified health efforts to alleviate zoonotic disease transmission. Furthermore, Karatu district experienced an outbreak of human blood for the first time in 1996 and involved 186 clinically suspected cases with 12 equivalent to 6.5% deaths. This outbreak affected 6 villages, which are Troma, Ayarabe, Rotiakati, Kimatembo, Gambia Simba, and Slahoma, and was allegedly caused by transportation of infected materials from Shoto, eastern Tanzania, where the disease had been prevalent since 1980, and from where fruits were being transported to Karatu and elsewhere in the country for sale. A similar outbreak occurred in the same area in 1997 and involving 11 cases or suspects, with 2, equivalent to 18.2% deaths. Hence, according to Kilonzo and others, in the year 2006, a survey was conducted in July 2004, whereby commensal and feed Lawrence and wild small carnivores were live trapped in 5 villages of Karatu district that had a history of plague outbreak, and 1 settlement in Ngorongoro conservation area near to Karatu district, Tanzania. Blood samples were taken and serologically tested for plague. Fleas were collected from the examined animals and from randomly selected residential houses. A total of 241 rodents, 1 Crossjura species, 43 dogs, 12 cats, and 4 slender mongoose were involved in the survey. Of the rodents, 14.5% were infested with fleas, which comprised of Xenophila brasiliensis, that is 45.8%, and Dinopsilus lepusus, that is 54.2%, with an overall population density of 0.2 fleas per animal. So 31, equivalent to 72.1% of the dogs were infested with the fleas, of which were Tenofaridae species. 35, that is 33.3% houses were infested with fleas, whose population was composed of Tenospherides species, Prorex irritans, Tunga penetrans, and Echinophaga gallinaceae. Infected rodents were found in all the villages, while the infected dog was found at Rotia Cat. 19, equivalent to 11% of the rodents, and 2, equivalent to 2% dog, harbored specific plague antibodies. It was broadly concluded that sylvatic plague was endemic in Karatu district and in Ngorongoro conservation area, in that outbreaks of the disease can occur in the area anytime if and when relevant conditions become favorable. Furthermore, McCurry and others in the year 2015 studied effects of land use on plague Yersinia pests, activity in rodents, whereby rodent trapping and pathogen sampling was carried out in their 3 sites in Tanzania. The Tronga and Kambi Yanyoka, which are situated at approximately 1,700 meters elevation and located within conserved area, and along the border of the Ngorongoro conservation area, that is cultivated area, and the Manyara site, that is situated at approximately 1,000 meters elevation and located within a conserved area and along the border of Lake Manyara National Park, that is cultivated area, estimated abundance of seropositive rodents near doubled in agricultural sites compared with conserved sites. This relationship between land use type and abundance of seropositive rodents is directly mediated by changes in rodent and free community composition, particularly by an increase in the abundance of the commensal species. Mastomys natarensis in agricultural habitats. Hence, these results suggest that land use change could affect the risk of local transmission of plague and raise critical questions about transmission dynamics at the interface of conserved and agricultural habitats. These findings emphasize the importance of understanding disease ecology in the context of rapidly proceeding landscape changes. However, since then, there is no any other study of the same kind that has been conducted in the area. In small mammal, long-term study is being conducted at our center using a capture marker capture CMWR will help us answer the following questions. Question number 1. Spatiotemporal species composition, age structure and sex ratio. Information on rodents community species composition is important for conservation and management of rodents. So this is because each species occupies its own niche, and at the same time, conservation of a given species depends on the maintenance of its niche. It has been reported that human interference and grazing pressure have influence on the rodent community species composition. On the other hand, it is well known that coexistence of several species in an area depends on their ability to partition their niches. Moreover, the age structures and sex ratios of rodents are important ecological characteristics for conservation and management of rodents. For example, potential of reproducing new individuals decreases with increasing age, which leads to skewed sex ratios and may bring negative demographic consequences in a given population. Question number 2. Small mammal population ecological characteristics over time and space. Communities of rodents are made of populations whose essential ecological characteristics can be described by their population dynamics, breeding patterns, home ranges, survival rates, and recruitment. It has been found out, for example, that densities of two rodent species, Permiscus astecus and Retrodontomys fluvensis, were influenced by different habitats and seasons. Also, Massawe and others in the year 2012 reported that reproductive patterns of grassrat Aphicanthus pneumoniae is seasonal depending on rainfall patterns, while Mriashimbe and others in the year 2019 reported a continuous production in M. natarensis in semi-arid areas due to availability of food and moisture. Whereas Boremans and others in the year 2014 found that home ranges of female adults of M. natarensis increased with population density, while those of males and subadults correlated negatively with it. Furthermore, Murungu and others in the year 2016 indicated higher rodent recruitment of M. natarensis in rice fields compared with farrowland in irrigated fields. Mriashimbe and others in the year 2019 indicated that survival of M. natarensis in semi-arid condition differed between dry and wet seasons and was lower in clay soils during the wet season compared to sand soil. Question number 3. The influence of ecological factors on distribution and abundance of rodents. According to Rebuga and others in the year 2003, rodent species distribution and abundance across landscapes are influenced by ecological factors such as soil types, nature and dense of vegetation, climatic conditions, disease, predation, and habitat exploitation by humans. Hence, absence of sufficient food and ground cover largely determines the number of individual rodents in certain areas due to less availability of food and increased predation risk. Moreover, bush vegetation provides safe sites for germination and growth of herbaceous vegetation, hence enhancing the diversity of rodent food resources, which in turn attract more of these animals. Therefore, this might lead to diversity of rodent species due to habitat variability caused by supply of different resources. Question number 4, genetic diversity and fitness. According to Gibbons and others in the year 2020, natural populations face a number of threats, including habitat loss and alteration, direct mortality from exploitation, invasive species, emerging infectious disease, pollution, and climate change. These threats are pervasive and global, so that an estimated 1 million species of plants and animals are at risk of extinction within the next few decades. Threats to wildlife populations often act synergetically, and genetic factors are central to the challenges confronting wildlife. According to Ceballos and others in the year 2017, loss of genetic diversity and inbreeding due to population declines and fragmentation can reduce population fitness directly, but also can reduce a population's ability to adapt to novel conditions produced by invasive species or climate change. So genetics and genomic concepts and the ability to efficiently study genetic factors in nature are important for quantifying and mitigating threats to wildlife populations. Over the past decade, the field of population genomics has developed across non-model organisms, and the results of this research have begun to be applied in conservation and management of wildlife species. Body parts obtained from trapped animals can help in genetic analysis of the same. Lastly, since many small mammal species, especially rodents, are human crop pests, non-invasive techniques can be used to determine dietary habitat patterns exhibited by those animals. Hence, several questions can be answered, which include food categories of rodents between different seasons and habitats. Food sources are one of the important aspects that are essential for existence of any living organism. Knowing food categories of coexisting rodent species is important for establishing their diet, need, breath, and overlaps, which describe their food resource competition and or partitioning status, and hence acting as a means for proper conservation and management of these animal species. So Gadisa and Shundera in the year 2015 reported that four rodent species, regardless of the proportion of difference, consumed plant and animal matters. These findings are incongruent with the ones by Addis and Becker in the year 2013, who reported that 11 rodent species consumed matters and arthropods, but in varied proportions. Furthermore, Murung and others in 2011 reported that the alternate breath of Gibiliscus vicinus was greater than that of Mastomys natarensis. These rodent species overlapped considerably in the food items consumed, ranging from niche overlap of 0.77 to 0.89. Grains stroke seeds featured high in the diet of M. natarensis, while plant material occurrence was high in G. vicinus, contributing to differences in diet partition, which may in turn facilitate their coexistence in farmland. Such information is not available in Karatu district, where people also cultivate various food and catch crops. Thank you so much for your time. My contacts are tel plus 255 0768 732 006. That is my WhatsApp number. And the other number is plus 255 784 802 565. My email addresses are jegidus@yahoo.com. The other one is jerwebuga@gmail.com. And the last one is irwebuga@fieldstudies.org.